All right, so I was saying I'm glad some talks this morning have done some of my prep work for me and explaining some of the concepts to you, which should make this go a little faster. But basically what I'm going to be talking to you about today is a energetic modeling approach to understanding habitability and microbial distribution in hydrothermal vent systems. So we all talk about habitability in astrobiology a lot, but habitability is something that we need to be able to quantify. And when we're talking about microbial organisms, we need to have a way to quantify environments that is not based on our preconceptions about habitability. For instance, we need to be able to decide whether this nice field with the sheep or this black smoker hydrothermal vent is objectively and quantitatively habitable in different ways. And the way that we do this in my project is we look at energy availability, because energy availability is a neutral determinant of habitability. We're not thinking about, we're not comparing temperatures, we're not just comparing the amount of energy available to organisms and the amount of energy they need to survive. So the environment that I look at are deep sea hydrothermal vents. We had a couple of talks yesterday that talked about very alkaline hydrothermal vent systems and ones that existed in sort of Archean oceans, but I'm looking at modern ones, black smoker vents. So briefly, for those of you who aren't familiar with deep sea hydrothermal vent systems, basically what happens is seawater is drawn down and percolates through the crust where it becomes very hot, very reduced and very metallic. And then it flows back up as hydrothermal discharge, and it also mixes with seawater, which has only been drawn down a little way into the crust, and vents both as black smoker hydrothermal venting, which creates this black hydrothermal plume, where the hot, reduced metallic fluid meets the cold, oxidizing metal-poor seawater, and iron sulfides precipitate out. But it also vents as diffuse fluid at 2 to 50 degrees when it's been mixed with a lot of seawater. So obviously everything that comes up there is being sterilized, but microbes that live both in the vent community and the subsurface can be sampled by sampling the diffuse venting where the fluid's a little cooler. And the key thing about these systems is primary production is chemosynthetic. So it's not based on photosynthesis, it's based entirely on organisms, microbes, which are making their living off the chemical redox potential between the cold oxidizing seawater and the hot reducing hydrothermal fluid. And on the right is a black smoker chimney, so you have a pretty picture. Now, at hydrothermal vent systems, geochemists, who are not me, can model energy availability to microbes for a number of different microbial metabolic strategies. So this is an image from McCollum and Chock, which is a well-known paper doing this, and essentially what they're showing you is over a range of temperatures, the energy available, and they're measuring it in calories per kilogram of vent fluid for a number of different metabolic strategies, methanogenesis, iron reduction, sulfur reduction, and so forth, changes over temperature. So essentially, at lower temperatures, oxidation metabolisms are favorable, and at higher temperatures, thermophilic and hyperthermophilic ones, reductive metabolisms are favorable. And they have also looked at it over a vent chimney, so this is meant to be a cross-section of a vent chimney, and they're predicting where different organisms would live based on the temperature within this vent chimney, assuming that you have the same geochemical fluid available throughout the vent chimney. So essentially what they're saying is you can predict what should be living where based on the geochemistry and the temperature. However, to understand what should be living where based on energy availability, we also need to know about energy requirements. So essentially there are two types of minimum energy requirement that we talk about in microbiology. They're both broadly called maintenance energies, but more specifically we have true maintenance energies, which is the energy required to maintain cellular function without growth. So this is like maybe an organism in the permafrost, it's maintaining its osmotic balance, it's repairing its DNA, but it's not growing. Growth energy, on the other hand, is when an organism over time is able to both do all that work of maintaining itself and reproduce. So these are two different types of minimum growth requirement, and one sustains the organism and one allows the community to grow. Now, maintenance energies also have a relationship to temperature. So maintenance energies, and here I'm talking specifically about the minimum maintenance requirement, not both of them, are measured in energy per cell per time unit in these sort of studies. So different studies will use different values for the energy and the time unit, and sometimes they'll use gram of carbon instead of cell, but it's basically energy per biomass per time unit. And this is adapted from Tihui et al., which is the best known meta-analysis of maintenance energies in microorganisms. And what it's showing us is, so they're doing kilojoules per gram of dry weight per day for maintenance energy, and they're graphing it over temperature. So you can see first off that maintenance energy rises with temperature in this meta-analysis of measured maintenance energies, and secondly that aerobic maintenance energies are slightly higher than anaerobic. However, there's a couple of problems with this graph for the purposes of deep sea hydrothermal vents, which are that first, if you see the little green circle, those are the only two autotrophic anaerobes on the graph. Everything else is heterotrophic. And secondly, if you look at the big blue circle, almost everything in this meta-analysis grows between 20 and 40 degrees. If we want to look at thermophiles and hyperthermophiles, the anaerobic organisms using reducing metabolisms at hydrothermal vents, we basically only have those two dots in the green circle as data. So we want data that allows us to confirm whether this trend that Tehus is looking at actually exists for the organisms we're interested in. Furthermore, this graph on the right is from Holler 2004. This is trying to look at methanogenesis, and here they're taking maintenance energies, basing them on the theoretical production energy required to produce ATP, but you can see that Holler is giving us maintenance energies from -20 to 130 degrees, which makes sense because of the boundaries of life, right? Except that we don't actually have culture-based data from microorganisms for anything like that. So that's pretty much entirely theoretical. And what would be really nice is if we had data that was actually based on growing organisms and measuring their maintenance energies to put into the geochemical models to predict where these things are going to live, rather than just using a theoretical piece of data. So this is basically my entire PhD, this study, and what we want to do is we want to obtain culture-based maintenance energy values by growing the organisms in pure culture in the lab. We then want to examine the distribution of the organisms that we're measuring maintenance energies for at hydrothermal vent sites and compare the predicted and actual habitability. So give our values to geochemists, get them to model where the organisms should be, and see if we can then see that in data we get from the field side. So the model organisms we're using are methanogens. This is primarily because methanogens, at least the ones we're looking at, are a one-trick pony at thermophilic and hyperthermophilic temperatures. The only thing our methanogens are doing, and this we're looking at a restricted group of methanogens, they are taking hydrogen, they are taking CO2, and they're producing methane. They're not using acetotrophy, they're not using any of the other weird methyl groups or anything, they're doing one thing. So it makes modelling their energetic availability very easy because you only have to look at one reaction, that one specific methanogenesis reaction, instead of alternative metabolic strategies they might or might not be using in field conditions. So these two graphs are my growth energy data for two groups of methanogens. The ones on the left are thermophilic methanogens, the methanothermococci. The ones on the right are hyperthermophilic methanogens, the methanocaldococci, which grow above 80 degrees. The thermophiles grow between 50 and 80 degrees, although these ones on the left all grow between about 50 and 60 degrees at their optimum temperature. Now these are growth energies, not maintenance energies. This is sort of preliminary data, so we're measuring the energy that they use when they grow instead of just maintaining themselves. But you can see immediately that apart from this one organism, both the hyperthermophilic methanogen and the thermophilic methanogens, these two graphs have the same scale. They have roughly the same range of growth energies. So their growth energies rise with temperature as they're grown at different temperatures, but they're not hugely different, even though this organism has a very different optimum temperature, which is different to Tihui, where organisms with higher optimum temperatures had higher maintenance energies. So the next thing I want to do is measure the actual maintenance energies, and the reason I'm doing that second is it's more difficult. You have to grow the organisms in continuous chemostat culture, which when you're working with anaerobes grown in salt water at 80 degrees is somewhat difficult because they don't mass produce chemostats to do that. So this is a graph from, I forgot to put the attributions, I'm a terrible person, and this is a graph from Holler and Jorgensen 2013, where they're showing that what a continuous chemostat is doing. You basically got an inflow, stirs, there's an outflow, and that maintains the population in the chemostat at the same number. So if you measure how much energy they're using, then you can measure their minimum maintenance energy because the number of organisms in the chemostat never changes. It's, so even if some die and some grow for the total number of organisms you can average out a maintenance energy. This is from Fardot 1987, where it's showing data where they're doing the same thing. They've got dilution rates for the chemostat, they've got a production rate, and they're graphing it out to get a maintenance energy measurement. So this is what we want to do. We want to grow organisms in a chemostat under certain conditions at a bunch of dilution rates. We should be able to graph it and then get a maintenance energy from the intercept. However, this is, by the way, this is the chemostat I use, and I just wanted to show you that because we can't buy one of these, I have jury-rigged it from things we had lying around the lab. It's a big Pyrex bottle, which we put a heat jacket on. This is actually a pre-built 2-litre bioreactor, and then we hook them up with pumps and a pH thing and just hope none of it falls apart or breaks, and it hasn't so far, so I'm very relieved. But this is the data I've got from my chemostat runs. Now, I've only been doing these for 2 or 3 months, so there's going to be a lot more data where this comes from, but you can see immediately that I'm not really getting that same neat slope that they got. Now, admittedly, I've been growing them at different hydrogen concentrations in case that has an effect on the amount of energy they're using, but it's pretty scattered. There's not really a neat slope, and I think why this is if you look at my hydrogen concentrations, they're pretty low. They're, micromoles per litre. And these are produced by gassing it with between 30 and 90 mils per minute of hydrogen. If you go back to Fardot, they don't have it here, but they're using 600 or 700 mils a minute of hydrogen, just truly immense quantities. And I think what's happening is even though my hydrogen rates are comparable to what you would see in a hydrothermal vent, it means that the organisms are not, in the other studies, the organisms are maxed out. They have all the hydrogen they could need, whereas at mine they might be restricted by hydrogen, and that's why I'm not seeing the same relationship. So we're going to have to work on basically finding out ways to produce the same data that other people had produced. But the important thing right now is the system is up, it runs, I can measure thermophiles and hyperthermophiles producing methane under these conditions. So secondly, we're also looking at collecting field data to make the comparisons once we have the maintenance energies. We collect field data from Axial volcano. It's off the coast of Oregon and Washington on the Juan de Fuca Ridge. We use JSON, the remote operated submersible. JSON has a fluid collector attached to it, which it sticks into diffuse venting sites to collect fluid that we bring up to the surface to do culture dependent, and our collaborators do culture independent abundance and diversity analyses on the microbes that we find there. So briefly, this is from our field data from our trip last year. We found that on the western side of the Caldera, which has a low hydrogen concentration, there were no Methanogens, but there were thermophilic and hyperthermophilic heterotrophs, so there are things living there. We just didn't find any Methanogens. On the eastern side of the Caldera, where they have higher hydrogen concentrations, we found Methanogens at both hyperthermophilic and thermophilic temperatures and fewer heterotrophs. So we are seeing initially that there is basically a hydrogen concentration relationship between Methanogen presence and Methanogen non-presence, which is consistent with data done from previous studies at this site. So that bodes well for our habitability studies using the specific maintenance energies. In terms of future work, we want to do a bunch more sampling at Axial. We're still waiting on data from our collaborators for the molecular data to look at both the metagenome and metatranscriptome and the geochemistry so the modelers can get to work. I'm going to be spending the next 2 years basically getting the maintenance energy data and sorting all that out. And thirdly, I have a master's student working with me who's going to start looking at thiosulfate reduction so we can also move this modelling to other metabolisms apart from methanogenesis, because we know there are thiosulfate reducers, thermophiles at these vents. We've isolated them from there. He can work with ones we've isolated from the vents. So I have hopes that that will produce some interesting comparisons with the Methanogens. And finally, I would like to thank my financial sponsors, everyone in my lab, and our collaborators who make all this possible by getting us the rest of the data. Thank you.
So do we have any questions for Lucy?
Just a quick question, are those thiosulfate reducers one-trick ponies?
No, they are not one-trick ponies, but we intend to grow them in conditions where they don't have the other things to reduce. So mostly those organisms reduce thiosulfate, also sulfur, sulfite, and a couple of other sulfur-related things, but we only give them thiosulfate.
Hi, great talk. I was just curious if, in addition to looking at the maintenance energy, you've put, or have been thinking about looking at also some of the other parameters that go into modeling, like half saturation constants for nutrients, or are you just focusing on maintenance energy for right now?
No, we're also looking at the half saturation constants, and I don't have the graphs ready yet, but we're working on taking the data we've got and putting into Michaelis-Menten and Lineweaver-Burke plots and looking at those things, absolutely.
Any other questions? Okay, let's thank her again.